I'm going to tell you a little bit. Actually, I'm going to try to tell you a lot about microalgae and what kind of things you can do with microalgae. I'm going to give you some examples that I've been involved with of production of nutraceuticals from microalgae, human therapeutic proteins, and also bioremediation. I've added that to the title over the last few days because I thought it was something very interesting that I wanted to share with you. The outline of my talk is, first I'm going to give you a little bit of introduction to microalgae, what it is that they are, what it is that they do, how you get them growing. I'm going to also give you a little bit about the history of microalgal cultivation, and I'm going to make the point that the history of microalgal cultivation has had to do with the development of scaling up technologies. That's always been a bit of a problem. And then I'm going to give you some examples of the products that you can get from microalgae, like astaxanthin, a natural product, a carotenoid, other high-value products like monoclonal antibodies with obvious applications in medicine, and finally, environmental bioremediation, how microalgae can help us take up CO2 from smokestacks and make that into biofuels. Microalgae are, first of all, very diverse. There is upwards of 30 to 50,000 species of microalgae. You can find them just about everywhere, mainly wherever there is water, and it doesn't have to be free water. It can be just a little bit of humidity, maybe even in your bathroom. They can grow very, very fast, a lot faster than higher plants, which makes them very attractive to produce even low-value products such as biofuels. They are, for the most part, unexplored, and the reason for this is that they are not very, very easy to cultivate, especially large scale. Like I said before, there are maybe 30, 50,000 species. Not all have been isolated and identified. Out of those, there is maybe 2,000 to 3,000 different species in collections around the world. Out of those, there is maybe a few hundred that have actually been studied, their physiology, their composition. And out of those, reducing even more, there is maybe a handful, certainly less than a dozen, that have actually been grown at commercial scale. So lots of them, very few actually grown up to large scale. And because they are unexplored, this makes them also a very valuable source of new substances. Now, what do you have to do to be called a microalgae? Microalga or microalgae is not a phylogenetic term. It's more of a functional term, if you will. You have certain characteristics, you can be called a microalgae. The first thing that you usually have to do is you have to be very small. Otherwise, you would not be a micro. But you can be a bacteria or bacteria-like. You can be prokaryotic, or you can be fairly advanced. You can be eukaryotic. For the most part, you will be unicellular, but you don't have to be. Some microalgae are actually clumps of very, very similar cells. For the most part, you will be very colorful, and that's because you would have a lot of photosynthetic pigments, chlorophylls, carotenoids, phycobilioproteins, and so on. Most likely, you would be aquatic. You would be a free-living organism, but not necessarily. Some algae live in soil. Some algae live in sediments, for example. Some algae live in statues. And for the most part, you will be a photoautotrophic organism. That is, you will make your own food from sunlight, CO2, and salts. But some also do have the ability to take up simple organic molecules and survive of those things like acetic acid, sugars, and whatnot. So very diverse. And what I'm going to do now is I'm going to show you just a few photographs of some of the microalgae I've worked with. Some of them I don't know what they are. I've isolated them from the field. I've never been good enough to identify them. But sometimes that's not important. What you want to identify is the product they can make. Some of them are very small. For example, on the left photograph, you have a little 3-micron cocci, little balls, basically. You have a chlorophyte on the right-hand side. It's about 8 microns. Just to give you a little bit of an idea of diversity, you have things like Xenodesmus. These are all isolated from Hawaii, by the way, where you have about 8 to 10 micron cells that actually clump up or live together in little 4-cell chains. You have also things like Botryococcus. Botryococcus is an organism that can make a lot of oil and is one possible organism to use for biodiesel production. That one actually makes big, big clumps of cells. Finally, another example, Lyngbia is a cyanobacteria. Lyngbia is a cyanobacteria which can get to be very, very large. These trichomes you can actually see with the naked eye, and they look almost like little balls of hair, and they're actually very strong. If you can imagine that scaling up of some of these cultivation of these cultures, when it's time to harvest the material to actually extract whatever high-value product you want to put out, morphology will be, for example, very important if you're going to use something like filtration, centrifugation, and whatnot. So some of the things that you have to keep in mind when you're trying to develop a product, for example. Like I said before, the very diversity of microalgae and the fact that they are largely unexplored is what makes microalgae very attractive in biotechnology. Some people will say, well, why not just go the synthetic route to produce some of these chemicals? There are actually two other big advantages to microalgae and to natural products in general. First of all, natural products have been a consistent source of new drugs. Estimates of 30 to 60% of new drugs coming to market over the last 10 years have their origins in the natural world, not in something just put together in a lab. Products that are of natural origin also have some other advantages. The fact that a living cell made them means that those chemicals already have some characteristics that would make them more useful to medicines. For example, solubility. Again, if it's been made inside the cell, it probably is soluble inside the cell. It can probably go through membranes, those sort of things. Those are characteristics that in a laboratory you would have to engineer into a synthetic molecule. So again, some of the work has already been done for you. As I told you before, I'm going to touch up on the issue of a scale-up a couple of times. The problem with microalgae is if you go out there, unless you happen to run into the spring phytoplankton bloom or a red tide, for the most part you do not see them. They're very low abundant organisms for the most part. And whenever you're trying to do some discovery, you're going to need more material that way you can possibly find in the wild, in nature, if you will. And especially if you're looking at industrial applications, you're just going to have to scale up. And what I want to do is I'm going to give you a little bit of the history of microalgal cultivation, and again, make the point that it has been basically a race to scale up cultivation. If we look at when did people actually start using microalgae in a non-biotechnological way, if you will, you can imagine that people in coastal communities in very ancient times were already probably consuming microalgae. Every time you eat a clam, you're probably eating some microalgae that clam harvested from the ocean. If you look at writings of over 2,000 years old, in China, they were already consuming nostoc, for example. When the Spaniards got to Mexico in the 1500s, they already found the Mexicans eating spirulina from Lake Texcoco. The same thing before the 1900s, when French explorers were in North Africa, they were going through Libya, Chad, looking for petroleum resources. They found people also consuming spirulina from Lake Chad. So people were already consuming these organisms and oozes without really knowing what they were. Maybe in 1673, when Anton van Leeuwenhoek actually started to look at these organisms, people finally, at least he did, became aware of some of these little animals swimming around in little drops of water. But he did not really know what he was looking at. Now, in the 18th, 19th century, there were a lot of studies on plant nutrition. People figured that plants needed things like nitrogen, phosphorus, potassium, and so on. And finally, towards the end of the century, of the 19th century, Van Minting actually started to do the first studies on microalgal nutrition. And what he realized is that microalgae needed the same kind of elements that higher plants needed. In 1890, Beijerinck was the first person to actually be able to cultivate a microalgae in pure form, chlorella. And in 1903, Czech actually started to, using those techniques, was able to specifically look at the nutrition and growth metabolism of microalgae once they had been able to isolate them and cultivate them in pure culture. In 1905, the first cultures of microalgae were actually started. That was in Plymouth in the UK, very small scale, test tube scale. By the 30s, at Woods Hole, you had people putting together 5-gallon reactors. Again, we're scaling up from test tube size to carboy size, if you will. In the 1940s, they developed the first automatic reactors, things like chemostats, or what we call today chemostats. In the 50s, Berlew actually published a book in which the first reactors of thousands of liters were described. And also was the first person to actually start putting some economic models to microalgae culturing. And the conclusion of those studies was that one could actually use microalgae, produce proteins, for example, to feed people who didn't have beef, and actually make money doing this. It was economical. Following that, there was an explosion, if you will, of groups trying to grow, again, larger and larger cultures of microalgae. And what was developed is what's called open pond technology, and I will show you some examples of that, in which you could actually cultivate many thousands of liters of culture at a time. Two main types of technology were developed: paddle wheel raceways in Germany, in Dortmund, and round ponds in Japan. And again, I'll show you some photos of that. Oops. Now, the interesting thing is, from the 50s to the 70s, a lot of people tried to cultivate a lot of different species for different products, and just about none were successful. As of the 70s, only 3 species were commercial. And I'll tell you about them in a little bit, but those were chlorella, spirulina, and Guna Lila, and I'll tell you why. And in the 1990s, what you actually have is, that's the company I used to work for, Microalgae. We created the first large-scale, actually enclosed photobioreactor, which actually allowed us to grow new species of microalgae, one of them being Hemerococcus for the production of Astaxanthin, which I will tell you about as well in a little bit. Now, the interesting thing about microalgae is they can give you all these different products, but it's actually fairly expensive to grow microalgae. So unless you're going to produce a high-value product, you'd probably rather put your money somewhere else. What the company that I'm working with now, Greenfield of Cambridge, what we're trying to do is develop a photobioreactor that is actually very inexpensive to run, which will allow us to grow microalgae for low-value products, but very large markets, such as biodiesel, biofuels. Now, just to show you some photos of what the different scales mean and what you can do at those different scales. On the top left corner, you have a bunch of different test tubes with a bunch of different strains of microalgae. That's about where the industry was at the beginning of the 20th century, if you will. Little test tubes of algae. From that, you can scale up to things like Erlenmeyer flasks, where you can maintain your cultures, but you cannot really do a whole lot of experimentation. Some experiments you can do, and the bottom right slide shows just a very simple temperature experiment, where we're trying to figure out this organism, what kind of temperature range did it have. And we found out that, well, this one specific bug would not grow at 15 degrees Celsius, but would grow fairly well at 20 and even better at 25. Again, small scale, relatively small experiments. Then you can move on to something like a chemostat. Actually, this is a pH stat, a 3-liter system, computer control. You can have very, very good control of your cells, maintain a constant physiological status of the cells, but you still have relatively a small volume. If you want to actually produce something, this is not enough. What can you do is you can start multiplying some of these small-scale units. These are 20-liter carboys. But again, you're trying to grow microalgae. You need to give them light. At some point, it really does not make any more sense to do this indoors. At some point, you want to go outside, large units, and use the free sunlight. And that's what you do. You go outside and develop photobioreactors that have a number of requirements. One of them is you have to try to control light. And outdoors, it's a lot more difficult than indoors. Outdoors, you have the sun, and whatever the sun sends that day down to you. But you can still, if you need to lower the light, you can always do that. You cannot increase the light. You need to control the temperature. Some organisms do have fairly tight temperature optima. Control the pH, both from a chemistry of the medium point of view and also from a CO2 nutrition point of view for the algae. You need to control the nutrients. Turbulence perhaps the most important one, because turbulence will determine how well you can mix the rest of the characteristics, the temperature, the CO2, the pH, the nutrients, and so on. And finally, you need to control the pests and the weeds. Some of the organisms that you want to grow are not the fastest growing algae. So any other weed that gets in there will overtake your cultures. So again, you want to protect that at very large scale. The characteristics that these photobioreactors need to have, and from a commercial application point of view, first of all, you want a system that will give you high aerial productivity. The reason for that being that some of the costs of a large plant are associated or related to the actual surface area that you cover, just land costs, for example. You also want a photobioreactor that will have high volumetric productivity, because some of the costs will be scaled to how much medium you actually have to handle, like centrifugation, pumping, for example. You want units that are of large volume, because some of the costs will be associated with the number of units, like, say, the number of pH probes, for example, computer nodes. You want a system that's fairly inexpensive, especially as you go into relatively low-value products like biodiesel. You want to be able to control the culture parameters, excuse me, and you want the system to be reliable. And you really have two options. You can go to open ponds. Some of these can get real large, or you go with enclosed photobioreactors. Now, the open ponds are real easy to operate. They're relatively inexpensive, but you have very little control over the conditions just because you are open. And because of that, their open ponds are really limited to those 3 species that I told you about that were developed up to the 1970s. Guna Lila, you can afford to grow in an open system because you can grow it in a very salty medium. So salty that anything else that goes in there will not survive. So that you can afford to have in an open system. Spirulina, a cyanobacterium, you can grow in an open system because you can grow it at a very high pH. It's so high that anything else that goes in there will not make it. And the last one, chlorella, is grown in open ponds, but only thanks to the fact that it can be fed sugars, it grows very, very fast, and it itself is a weed, so it will tend to outcompete everything else. And also, outside cultures of chlorella need to be supported with clean lab cultures continuously, just renovated continuously. If you go to an enclosed photobioreactor, your life becomes a little bit easier. You can really control the conditions in a photobioreactor. The 2 photos on the right are one of the systems that we used at Mera in Hawaii. The physical barrier helps you keep the contaminants out, so you can grow things that are not weeds, that are not the fastest growing bugs. And you can use that technology to grow many different species. During our Department of Energy project, we grew about half a dozen different species at very large scale that had never been grown before, again, because of the lack of these large photobioreactors. Some of the photos of open ponds. Top left are the ponds that we had at Mera Pharmaceuticals. The one on the top right is a pond from Cyanotic, which was right across the street from us. That's a 500,000 liter pond. Those are about almost 10 times as big as each one of the ones we had at Mera, very large ponds. On the bottom right, you have, again, open ponds. They are producing spirulina out of earth rice in Calipatria, California. And on the bottom left, you have open ponds in Australia producing Guna Lila for beta-carotene. Again, those ponds are the easiest thing in the world to take care of. They're so salty that, again, the only thing that will survive in there is Guna Lila. These are the kind of ponds that are being used in Japan and Taiwan for chlorella production. These are round ponds, and they have this big arm that kind of sweeps the whole thing to keep it mixed, as opposed to paddle wheels like the ones I showed you before. And this is another photo of Guna Lila and the ponds in Australia. You can see, especially in the top right picture, you see all that white stuff? That's not northeast snow, that's salt. That's how salty it is in there. And again, Guna Lila loves it, and nobody else does. Some examples of enclosed photobioreactors, again, outdoors. On the top left, you have a couple of examples from Italy of, first of all, tubular photobioreactors. Those are actually immersed in a pool of water for temperature control. It's something you could not possibly do to an open pond. The second one from the left on the top is a panel reactor, similar to the one on the far right. Those are from Israel. Here, what people are trying to do is use very thin films of algae under very, very well-controlled conditions. And on the bottom, you have, on the bottom far left, is a system, also a tubular system from, actually from southern Italy. And here what they're doing is they're trying to control the temperature inside the tubes by spraying them with water. And as that water evaporates, it actually cools the inside of the tubes. Fairly efficient if you're in a very dry place. The last one is something called the Biocoil. And I think, I'm not sure, that's a unit installed in Florida at, I think it's Harbor Branch. And I believe they're using it, or one similar to this, to produce algae to feed seed cultures of clams and bivalves and that sort of thing. For the most part, these are fairly expensive systems. Some other things people have tried. On the top right, you have actually what looks like an open pond. It's like a little pool of spirulina in southern France that's actually just covered with a greenhouse. Maybe not the most high-tech, but probably very inexpensive. On the bottom, you have another type of biocoil. That's an indoor biocoil. And on the right, what's called a biofence out of the UK. These are actually also fairly expensive systems made out of glass piping and things of this sort. Again, if you're trying to produce a very expensive product, this could work. For other products, it's just too expensive. And on the top left, what I consider a little crazy things people have tried to do, that is to actually have your reactors indoors, but have a solar collector outdoors. And what you end up doing is, unless you have a lot of collectors, which happen to be very expensive, you are actually capturing very little light in some kind of a dish like that you're then trying to spread over your whole culture. Really not a very efficient way to bring light in. I think this is the second to last photobioreactor slide. This is a very large system out of Germany. And here again, you see a lot of glass tubing on a biofence type distribution. And in this plant, what they're doing is they're using this algae to grow on CO2 produced by a cement factory. So this is actually capturing the CO2 from the factory. And then they're using the algae to put into cookies and pasta and breads and all kinds of things. I believe this is the last photobioreactor photos. Here you have 3 examples from Hawaii. The top left is what Cyanotic calls their phytodome, which is actually a paddle wheel-driven pond covered by a greenhouse. The photo in the middle is a relatively small, about 1 meter across, biodome that is used by Microalgae on the island of Maui to produce also Astaxanthin from Hemerococcus. And on the bottom right is Mera's growth module that I told you about earlier. Last, I'm going to show you a couple more systems that have been developed by Green Fuels, the company that I'm working for now. And what I wanted to show you here is one of the systems, the bottom left. Those are hard plastic tubes in Arizona hooked up to the flue gas from a natural gas burning power plant. So in Arizona, we are doing some feasibility studies looking at the flue gas coming out of the power plant being fed into the reactors. Another experiment or another feasibility study that we have going on right now is in New York, taking flue gases from a coal burning power plant, using those to grow algae in these simple bags. This is a relatively simple technology out of University of Maryland, and I don't remember the name of the lab right now. Now, the goal of biotechnology is to make money by developing marketable products. A lot of people say the goal of biotechnology is to produce these wonderful medicines or to save lives and so on, but that will not get you the money to get started. What's going to get you the money to get started is to show somebody you're going to make money for them. And that's where your board of directors is going to be asking for and so on. How do you do this? Okay, the first thing that you need to do is identify a desirable metabolite or a process, the microalgae, and the microalgae that produces the metabolite or runs the process. You have to establish a large-scale cultivation because at some point you are going to be limited by biomass. And finally, you have to market and sell the metabolite. I'm going to give you 3 examples that I've been involved with. Excuse me. First of all, I'm going to tell you about astaxanthin production from Hemerococcus pluvialis, a chlorophyte microalgae, production of monoclonal antibodies using Chlamydomonas, also another chlorophyte microalgae, and finally bioremediation. And I'm going to show you a few examples from the work that we did in Hawaii for the Department of Energy and a couple of things from the work that we're doing now at Green Fuels. Okay, Hemerococcus. It's a cute little green alga. It's about 10 microns across. And it has this ability to really change on you. If you're growing it under the right conditions of temperature, pH, nutrients, and so on, it is this cute little green alga that you see on the top, on the little corner picture, swims around. It's got 2 flagella, and it has absolutely no astaxanthin inside of it. It has, in a way, no value. Now, it turns out that you can stress it, and following the stress, it will do a few things. First of all, it will stop dividing. You can imagine that if your environment is going bad, maybe you do not want to have kids. In a way, the algae are doing the same thing. It also starts making a very tough shell around itself, and it starts accumulating astaxanthin. It's a very natural response. We do not need to put any nasty chemicals or poisons in the water to convince the algae to accumulate astaxanthin. It just does it. You take the nutrients away, increase temperature, some of those things. So it's a very natural response to the environment, to the changing environment. Astaxanthin is a very good antioxidant. It can be used not only to feed, for example, to salmon, so that the farm salmon that you buy has got this beautiful pink, orangey color. You can also take it as a nutraceutical. It's a very good antioxidant. It has applications as a photoprotectant, anti-inflammatory, anti-cancer, and so on. And I do have some reprints here, if any of you guys are interested, to pick up at the end of the talk on some of these applications. It also has a high retail value. You can buy this for the equivalent of tens of thousands of dollars per kilogram of pure astaxanthin. A nice product to produce with relatively expensive microalgae technology, if you will. There are 5 major steps to making astaxanthin from Haematococcus. First of all, you have to, again, scale up. Otherwise, you don't get enough. Use your photobioreactors. When back at Mera, we use both open and closed photobioreactors. Then you have to harvest the microalgal biomass, separate and recover the astaxanthin, and finally come up with an end formulation for your products. And quickly, we go through all of them. The way we scaled up at Mera, and the way you scale up just about everything, again, you start with some relatively small scale cultures, like the early Mare flasks on the left top graph, a picture. You scale that up into chemostats. Chemostats then will produce enough inoculum that you can take it into carboys, and then from carboys you have enough inoculum to go into an outside photobioreactor. We use the system that we developed, the Mera growth module. As I told before, we can computer control it to determine nutrient concentrations, the level of turbulence, gas exchange, temperature, and pH in the system. The units that we had developed were 25,000 liters, and they, at the time, they were the largest enclosed photobioreactors in the world. They're not anymore. The system in Germany is actually longer volume, and the one we're putting together at Greenfields is even larger still. And the whole point of these enclosed photobioreactors is to produce green biomass. Again, we're not producing astaxanthin yet. For that, we need to stress the algae. And we do that in an open system. Thank you. And what you have here is 5 ponds from Mera. The first one on the left is a pond that has just been inoculated, and you see that there is some fairly bright green biomass in there. The pond after that, or to the right of it, was inoculated a day before, the next one a day before, and so on. So you got 5 different age ponds. You can see that every time they get a day older, they become more and more red, and that is the accumulation of the astaxanthin. What you end up is with biomass that's about 2.5 to 3.5% astaxanthin of the dry weight, fairly rich in color. And in fact, this is what it looks like. To harvest it, the Haematococcus biomass is actually very easy because the cells have stopped dividing, they've kind of fattened up, gotten a little heavy, they have this very tough shell. All you have to do is stop the paddle wheel, as you can see on the top left picture, and the biomass just kind of settles to the bottom of the pond within half an hour. Then all you have to do is open up a valve, decant the top of the pond water, and the next morning you have somebody who physically goes out there with a squeegee and pushes all the biomass down the drain on the bottom left corner. That goes into the ground, through a pump, and into a processing building, which is represented by the tank on the bottom right. Now, what you have to do there is, specifically to this product, not necessarily to other products, like I said before, Haematococcus under stress, besides accumulating astaxanthin, has built these very tough shells. You have to break those. If you do not, you can feed that product to salmon or to yourself, and the astaxanthin will go right through you. You will not get any of the benefits. You'll end up with red stool, but that's about it. So we use a homogenizer to break those cells, 10,000 psi, and then that material gets dried to less than 5% moisture, and that's one of the systems that we use at Mera to very gently dry the product. You have to be gentle, relatively low temperature, otherwise astaxanthin being such a good antioxidant, that means it's also very reactive, and it gets oxidized very quickly, and you need to protect that. So you need to protect it from oxygen and from heat and from light. Once you have the biomass, you could in principle consume that, and some people think that it tastes kind of fishy. I kind of like it. But if you want to put it into pills, it's best to actually extract the astaxanthin, and that's what we did. And on the top left, you see a small sample of the oleoresin, the extract from the algae. That's probably about 6 or 8% astaxanthin by weight. And then that's what would be put into the final products. And that happens to be the Astafactor brand. That's what we made at Mara. It's very good for you, actually. And that's it for astaxanthin. A little bit of monoclonal antibodies. And this project was actually a collaboration with Rincon Pharmaceuticals of San Diego. And Scott Franklin, their chief scientist, they developed techniques of which I do not know enough, and you guys probably know more than I do, on how to put the right piece of genetic material into the chloroplast of a microalga so that the microalga produces a monoclonal antibody. They developed this technology with Chlamydomonas, and they were able to show that these Chlamydomonas cultures would accumulate or would produce these antibodies. The problem they had is they had no idea how to scale up microalgae production. So that's where the collaboration came to be. Excuse me. Antibodies are naturally occurring proteins that the body will make or will produce to fight disease, confer immunity to you. The way, excuse me, antibody therapeutics are made today are using mammalian cells. These are called CHO cells. I believe that stands for Chinese hamster ovary. I call them rat ovary cells, but just so people go for the algae instead of the rats. Anyway, these proteins can be produced by a number of different processes, if you will. They need to be purified before they actually get injected. They are, from a industry point of view, it's a very fast-growing segment. And there's actually, out of 250 therapeutic proteins in clinical trials, 150 of those are monoclonal antibodies. The problem seems to be that there is only so much capacity in the industry today to produce enough CHO cells to make some of these antibodies. That makes the process also very expensive. And down at the bottom, there is a table showing some of the products that are being produced in that way. Excuse me. So the problems that Rincon saw is it's going to take a while to scale up one of these processes if we go with something like mammalian cells, with the CHO cells. There is a long manufacturing scale-up period. It's actually a very expensive way to grow cells, and which ends up with a very expensive product. Now, cost of goods of $500 per gram. Before I was telling you astaxanthin, we could sell for a few tens of thousands of dollars per kilo. That's a few tens of dollars per gram. This is maybe 50 times more just to produce it, let alone what you would sell it for. This makes it very expensive to get into the business, if you will, the fact that they are so expensive. And at the same time, there is a lot of pressure to reduce costs. And Rincon felt that algae was the way to go. The advantages of algae is you do not really have to develop anything majorly new from a genetic point of view. You use technology that has been used on mammalian cells, on yeast, bacteria, and so on. And once you produce the protein, you purify it, again, you can use it as a therapeutic. The one advantage of microalgae also, and one of the reasons why they did not go, for example, with a bacterial cell to try to produce these proteins, is that bacterial cells do not have the proper machinery to give the protein the proper folding. And don't ask me anything about that. I just know that, but I don't know the details. So anyway, with microalgae, some of the advantages you have is they're expensive, but not so expensive to produce. They're fairly safe organisms, and they're also known as GRAS. GRAS stands for generally regarded as safe. We've been eating microalgae for many years, let's put it that way. I cannot give you a lot of the details of the results because this is pretty much under wraps, but just to give you a feel for what we did, this turned out to be a very small study, mainly because the Hawaiians, not just the local Hawaiians, but the people in Hawaii, the state, in the end, did not give us the proper permits to work with GMOs. So anyway, it ended up being a very short study, but we were able to demonstrate expression of 2 different monoclonal antibodies in microalgae under field conditions. The levels of expression that we got were actually higher than the levels of expression they were able to get in the lab. So actually, it's usually the other way around. The field is trying to match all the wonderful things you get in the lab. We actually showed them a trick, which was very nice. And we were able to do this within 2 months. That's how fast you can get it done with microalgae. And that's it for monoclonal antibodies. The last thing I wanted to share was some of the work we've done on environmental bioremediation. First of all, just from the point of view of capture and sequestration of CO2 from smokestacks, and later using some of that biomass to actually produce biofuels. The concept is simple enough. You use photosynthesis, the top formula, to make biomass and oxygen. And the diagram, again, shows some stationary combustion system, a power plant of some sort, producing CO2 and other flue gases. The CO2 can be used by the microalgae in a photobioreactor, and you end up with a bunch of algae biomass, real simple, from which you get two things. On the one hand, the sequestration of CO2, and on the other hand, the commercial products. They can tolerate quite a bit of noxious gases, mainly things like nitrogen oxide that you get in just about anything you burn, sulfur oxides that you get if you're burning things like coal, for example, biodiesel. Microalgae culturing can be kind of expensive, but that's okay. If you produce the right commercial products, you can actually pay for the sequestration and even make some money. And the whole process is based on renewable photosynthetic carbon fixation. So you have relatively minor impacts on the environment. The project that we did for the Department of Energy, one of the first things we needed to do, and is some of the data that I'm going to show you, had to do with selection of the microalgae for this process. We wanted to look for algae that were fairly temperature tolerant, and you can imagine that you have a lot of fairly warm flue gas going into an enclosed photobioreactor. You want the algae to be able to put up with that. You want algae that are fairly pH tolerant. You can imagine that if the power plant is under pressure to produce more or less electricity, it will produce more or less CO2, which will affect the pH of your system. You want, again, the algae to be able to put up with that. You want algae that will be able to put up with the NOx and the SOx in the flue gas. And finally, you want algae that will produce something besides just biomass that you will be able to use to offset the costs of the sequestration. And finally, you want to look at what the actual CO2 capture rates are under different conditions of pH, under different gas mixtures. And finally, I'm not going to have time to talk about it, also whether the algae can capture carbon into non-biomass fractions. And if anybody's curious about that, I have other slides that I'll be happy to talk about. One of the first set of experiments that we did was, again, looking at our collection. We look at about 54 different microalgal species and see which ones were more or less temperature tolerant. And I don't want you to remember the graph or anything, just to give you an idea that different species do have different temperature optima and very different growth rates. And again, the top right slide, just an example of a diatom grown at 25, 30, and 35 degrees Celsius, very clearly, you can tell very clearly which temperature does and doesn't work. Another type of experiments, these were done in chemostats in larger units, looking at the actual amount of CO2 that's taken up by the algae. On the top graph, that's photosynthetic CO2 uptake rate, milligrams per liter per minute, and at 3 different pHs, at 6.5, 7.5, and 8.5. And what you can see is that different species have different rates of CO2 capture, and that those are different at different pHs. And on the bottom, what I wanted to look at is, okay, I know how much CO2 the algae can take up. Now, from the point of view of efficiency, and in this case, efficiency is a little funny. The way I described it here is once you have CO2 dissolved in the medium, some of it is actually going to get degassed back into the atmosphere. Now, what percentage of that dissolved CO2 is actually taken up by the algae? And that's the result on the bottom. And then what I wanted to do is, well, what's the relationship between the two graphs? And here you get something really cool. So here I'm looking at, on the bottom, on the x-axis, the actual CO2 uptake rate of the algae. On the y-axis, the percent of that CO2 that is actually taken up by the algae as opposed to the percentage that's degassed. And what you have is that, yeah, different algae have different uptake rates, but the efficiency value is really dependent on the pH, independent of what species it is. This is significant from the point of view of how to manage an algae farm to take up CO2. Do you want it to be more productive or more efficient? And depending on where you are, depending on how much land you have, you may choose to run the plant at one pH or another. So just an example. The other thing that we wanted to do is select algae by what kind of flue gases they were able to put up with. And this is a table showing, depending on what you're burning, whether a couple of different types of coal, natural gas, fuel oil, what kind of gases can you expect in the flue gas stream. Some of them have sulfur oxides, especially if you're burning coal. Some of them are a lot cleaner if you're using natural gas. But again, which algae can put up with what? And here is the results, same kind of graphs that I showed you before. On the top graph, the actual CO2 uptake rates. Again, different algae, different rates, under different gas conditions. And on the bottom, again, looking at that efficiency factor. Once the CO2 is dissolved in the water, how much of that goes into the biomass versus how much of that is degassed back out into the atmosphere, what inefficiencies you have. And what you have here is that basically all the points come down on the same line. So I did not find an effect of different gas mixtures on the efficiency of the system as I did with the pH, which makes me happy because I can go to a CO2 source, whether that's a coal burning plant, propane burning plant, whatever it is. A couple more slides just to show you some of the work I'm doing now with, or we are doing at Greenfield, and that is the power plant down in Arizona and the photobioreactors. One of the things that we want to do during a feasibility study is to show that the algae will survive the local conditions. That's why we're doing this work in Arizona or in Dunkirk, New York. And what you have here is just, again, just one little example of growth at Red Hawk, that's the name of the power plant, with grown on the flue gases, but on two different cultures. The difference being whether the medium is made up with local water, and there are some issues with water down in Arizona, or potable water. And actually, we are finding that there is a bit of an issue with the local water. Okay, some of the things that you want to do to see whether this is going to work at a specific power plant. Other things that we're doing, for example, in New York at the Dunkirk Power Station, what you have here is 3 different species grown, all of them on the local lake water. This is water from Lake Erie. And out of each species, 2 different cultures. One of them grown on clean, pure CO2. One of them grown on the local flue gas from the power plant. And what you see is that actually the 3 different species behave differently, but that's not dependent on whether they're being grown on pure CO2 or on the flue gas. Again, actually, that's very good news. And, okay, I am about out of time. So I'm going to leave it at that. Thanks, Sonia, for having me here, and thank the rest of you. And like I said, I have some papers out here. Some of them have to do with astaxanthin production. Some of them have to do with CO2 sequestration. Help yourself and ask me any questions.
Any questions, please? This microphone is just being projected here. That, you know, in other words, it's not going to sound like it's on.
Hi. I was wondering if the monoclonals that you produced, if anybody measured the binding efficiency or the affinities of them, and how they compared to those produced in show cells. Or
if anybody did, would have been the people at Rincon. We were not involved in any of that. And but Scott Franklin would be the person to contact. And if you're interested, I'll get that to you, that information. Yeah.
I'm BJ from Bristol Community College. My question is on the growth module. How do you clean up the system after the algae is harvested?
The system that we had in Hawaii, the tubular photobioreactors were made out of polyethylene, and they would stay fairly clean for about 6 to 8 weeks or so. Whenever they got, especially near the surface, they would get fouled out with algae. We would actually take the culture out of the photobioreactor, fill it up with a bleach solution, circulate that, clean them out, and then re-inoculate again. We usually were able to do that for about 2 to 3 cycles, at the end of which it was just too much crud on the top. And at that point, we would actually cut down the plastic, roll it up, take it to the dump.
The second question is, on the predator control, especially flagellates. How do you control the protozoans which can really chew up your mass production?
In our system, we never had a problem with flagellates. We used to have ciliates coming in. The way you control that is, first of all, you don't let them in. So you try to be, yeah, I mean, that sounds easy, but I mean, you try to be very, very clean. And at some point, if you end up with contamination, there's a couple of things that you can try, and I'll keep them to myself. But at some point, you actually have to just dump a culture. It happens.
Yeah, once upon a time, I tried allyl alcohol from garlic.
Mm-hmm.
Very micro doses seem to paralyze the protozoans and then allow the slow-growing algae, whatever you are culturing, to come back.
Yeah.
But that was a very drastic control mechanisms that I had to use. But garlic seemed to work a little bit, but not very long.
Okay.
Very long. It wears out. Volatile alcohols wear out very fast. So, thank you.
Thank you.
You mentioned that the permits didn't come through for your work on the monoclonals because they were GMO. Can you say a little bit more about what the regulations are, and can you release cultures into the environment, or do you have to sterilize them, things like that?
The regulations we had to work with were very specific to Hawaii. And in Hawaii, anytime you want to bring in an organism, you need to get an import permit from the Department of Agriculture. And that basically, you send in an application, it goes through a review process at 2 or 3 different levels, especially if it's something new. We did get a permit to import from the Department of Agriculture, and in fact, we did import a couple of different strains, if you will, of the microalgae from Rincon. As that was happening, the Department of Ag was sued by call them ecological groups who just wanted nothing to do in the state with GMOs. They didn't care what it was we were trying to do. They didn't care what kind of safety, what safeguards we were putting together. We did a number of experiments to show how easy it was to kill these cells if we needed to kill a culture. Some of the things we were questioned was, well, what happens if a hurricane comes through? How quickly can you kill the cultures before the hurricane actually spreads this all over the world? And we thought that we did the experiments and we put the procedures in place to calm any fears, but to be quite honest, and without trying to offend anybody, some of these folks are just anti-GMO, period. No matter why or what, or it doesn't matter. So the court stopped us in the end.
I'm Sang Yong from Ivy Tech Community College in Indiana. When you said you produced a monoclonal antibody much higher level than CHO cells, what kind of expression?
No, sorry, not than CHO cells, that the microalgae had been able to produce in the lab.
Okay. But you said the expression level was higher compared
in the
to
field than in the lab, yeah.
Okay. What kind of expression system do you use?
What kind of?
Expression system? Is it
You have to ask the
induction
Rincon folks. I do not know enough to give you a proper answer, sorry.
You also said you stress the cells when you produce the
Astaxanthin.
Yeah. What kind of stress do you apply?
We use a number of stresses. What you have is, let me just go back up real quick, just to put it into perspective a little bit. Okay, when you grow them in the photobioreactors, like I said before, you're trying to keep the cells real happy, which keeps them green and no astaxanthin. You give them all the nutrients, temperature, pH, everything that they need. And when you take them to the pond, what you would do is actually harvest a portion of one such photobioreactor, put that into a pond, and the first thing that you do in the pond is you add a bunch of fresh water. What that does, it immediately dilutes the cell concentration and the nutrient concentration. So that's the first stress. Nutrient concentration drops. The second stress is by diluting the cells, you don't have quite as much self-shading as you do in the enclosed photobioreactor. So all of the sudden each cell is getting a lot more sunlight. That's another stress. Those are the kind of things that tell the Haematococcus, hey, your environment is kind of going to heck. You better do something about it. And what they do is they go into a defensive mode. So they stop dividing, they make the little cyst, the shell around themselves, and they accumulate astaxanthin. And this is also very easy to do in the lab. You do not need to be in Hawaii in a lot of sun or anything.
I'm Todd Murphy from Ivy Tech Community College in Indiana also. With the problem you had with the GMOs, has there been any research into the development of non-wild type strains that would be oxytrophic mutants or something that would keep them from being able to propagate in the wild?
I have not been involved in any of that.
Okay.
I'm not involved in the project anymore, so I do not know what Rincon may have, how they may have decided to move forward.
Okay.
I do not know.
Hi, Tom Dietz from Lansing Community College. Since you've worked with a number of companies in Hawaii on all these projects, can you give us a sense of your workforce issues, particularly workforce issues for people who might be the folks we're training? That is to say, community college level folks. Did you find they had the training they needed, or do you have specific things that you felt were lacking?
For the most part, people that we hired, we had to train. Very few people came with much of any training. But part of it is probably due to the fact that Hawaii and Kona especially is fairly isolated. There are no programs, certainly on the island that I'm aware of, that do much from a biotech point of view. There is relatively few people that have ever grown microalgae as well. So for the most part, we had to train everybody we brought in.
Tom Burkett from the Community College of Baltimore County. I was just wondering if the cost difference that you saw between the astaxanthin production and the monoclonal was because, is there a lower standard of purity with the nutraceuticals versus something that's injected, and that might be a problem later on? I mean, you might not have that same cost difference versus...
That's part of it. There are two things. First of all, the level of purity needed. And second of all, also the concentration of the chemical of interest in the cells. When you have things like astaxanthin, 3-4% of the biomass versus monoclonal antibodies, I believe some of the proteins may have been in the fraction of 0.1% of the biomass, if I recall correctly. That already means you have to produce a lot more biomass.
Wayne Suits, Community College of Rhode Island. Relative to the production of the biofuel, was there a separation step involved in between collecting the biomass and producing the biofuel?
It depends on what biofuel you're going to produce. If you're going to do things like biodiesel, you would need to separate the oils, transform that. You can, however, also just use the dry biomass as a co-firing product, if you will, something you can mix back with coal and put into your power plant. So it depends.
If you were to separate it, would that be a chromatography step?
My understanding is that's not. Chromatography would be very expensive. There are other ways to separate oils. Yeah.
Craig Caldwell, Salt Lake Community College. The nice thing about them is they're photosynthetic, but what is the nutrient load that you have to put in there? What's the nature of those nutrients?
Well, again, it depends on what kind of product you're going to make. For things like astaxanthin or relatively high-value products, we've always used fairly clean chemicals, sodium nitrate, potassium phosphate, those sort of things. For very, very large-scale applications like what biodiesel production would need, we're looking for agricultural waste products even, things like manures to use as the nutrient source. Otherwise, you have also issues of, well, how much energy do you have to burn to make the potassium nitrate? Things that don't really make sense.
So I'd like to thank you again, Miguel. It was very interesting, as you can notice by the questions. And you're going to be around today and in a little bit, or sometime tomorrow too. So if you have any other questions, please, chat with Miguel. Again, thank you. And there's more food at the back for breakfast. And I believe the vendor show is just about ready to start. So I think this has been a wonderful start to the day, Miguel. Thank you very much for a really informative talk.
Thank you.
And we'd like to give you a shirt.
Oh, I love it. Thank you.